Welcome you all to Anjan Bios. Today in this video we are going to study about the most important post-fertilization event, the development of embryo. Embryogeny is the subtotal of changes that occur during the development of mature embryo from a zygote. It is of two types, dicotyledonous or dicot embryo development. Number 2, monocot embryo development. In this video, we are going to discuss about the development process of dicot embryo. Okay, let's start. You see, remember this structure. This is how double fertilization takes place. As a result of double fertilization, we have one zygote here. It is a result of generative fertilization between one male gamete and the egg. Then the second male gamete fertilized with the two polar nuclei become three. It will form the triple fusion, and as a result of triple fusion, we have one pen or primary endosperm nucleus, which later developed to endosperm, the nutritive tissue for this growing embryo or the zygote. Today we are going to study how this zygote transform to embryo and they differentiate to form the different parts of the dicot plant. Let's come to the class. You see, this is the structure of a zygote. You know, this is a diploid structure, and diploid cell, this one, is formed as a result of generative fertilization between one male gamete and the egg or female gamete. Then it formed a zygote. This zygote now going to differentiate and form the different structure of a dicot plant. What happened? In a typical dicot plant, first the zygote elongate and divide by a transverse wall like this into two unequal cells. The micropylar, the cell towards the micropylar end become larger, is called basal cell, and the cell towards the salazal end or antipodal side is called terminal cell. You see two unequal cell. This is called terminal cell and this is called basal cell. This terminal cell present towards the antipodal and it is also known as embryonal cell. This basal cell, this one, divides transversely. You see, in this way the transverse division takes place and produce a filamentous suspensor of 6 to 10 cells. You see this way. I showed in this diagram 6 to 10 cells here with a large basal one here, this one. And the first cell of the suspensor towards the micropyle is called haustorium. The last cell of this suspensor adjacent to this embryonal cell is called hypophysis. This hypophysis later give rise to radicle or root. You see, this is the actually this suspensor push this embryo, this embryonal cell or terminal cell towards the middle of the endosperm so that it get the proper nourishment from the endosperm. Then this embryo cell undergo two vertical division. They first form a 4-cell embryo here, okay, then it will form a 8-cell in this way. This is a 4-cell embryo and this is a 8-cell embryo here. This is called octant stage. This is called quadrant stage. This 8 cell here, they form 2 tiers. These 8 cell arranged in 2 tiers. The outer one is called hypobasal and the inner one is called epibasal cell. The 8 embryonic cell, these 8 embryonic cell divides peritoneally to produce an outer layer of protoderm. You see, this is the outer layer of protoderm. This one here, this is outer layer of protoderm or dermatosome. Finally, this dermatosome will form the epidermis. The inner cells, these cells are known as procambium and ground meristem. This will form procambium and ground meristem. This procambium give rise to stele or vascular strand. The ground meristem produce cortex and pith. The rate of the growth of the cotyledon is very high and initially this structure is a globular structure. This is called globular stage, this one, where the structure to some extent rounded structure is called globular structure. It is called globular proembryo. But later, with the growth of the cotyledon form from here, it becomes to some extent heart-shaped like this. You see, from to some extent heart-shaped, this one. Slowly, the rate of the growth of the cotyledon is very high, so that they become elongate tremendously in this way, while the plumule or shoot meristem remains as a small round of undifferentiated cell here. You see, this is the whole structure, and this root meristem is here, and this one root meristem, and this is the structure. Okay, you see, this is the fully developed structure. How this embryogenesis takes place step by step, I just want to show you. If you check the structure of a mature dicot embryo, you see is a embryonal axis with 2 cotyledon. Here 1 and 2, this is the embryonal axis, and this is 1 and these are the cotyledon, and the part of the embryonal axis above the level of cotyledon is called epicotyle. Here, this epicotyle terminate into plumule. Here, this is the plumule, and this is the part where fusar stems will develop. Then, the part below the levels of cotyledon here, this one is called hypocotyle, which terminate in the radicle. You see, this is the hypocotyle, and it terminates radicle. The root tip, this radicle, you know, this radicle is responsible for fusar roots. Then the root tip here, this root tip is covered by a root cap, which is called calyptra. So this is the structure of a typical dicotyledonous embryo. This is all about how this embryogenesis takes place within the dicotyledonous plant. Thank you.